it's yes. Is it good, or is it kind of,
Yeah, no,
lackluster?
Ooh,
it's a good turnout.
already 50 people.
And then like a bunch of attendees are also following this on the YouTube live stream. So we'll see a partial number here as well. But once it stabilizes, we can get going. It usually takes a couple of minutes. Okay.
Oh, I see my students coming.
Mhm.
Yeah, I was checking the same thing. I was checking in the students were here.
Okay, while we get started, I wanted to thank everybody and welcome Vanessa, Marco, and Lou to the organizing group for this webinar. Thank you for carrying on the torch, and also thank you, Professor Tuckerman, for being the inaugural 2021-2022 speaker. Now, I wanted to point you to two of the upcoming seminars in January and February. In January 28th, we'll have Gustavo Scuseria. And then on February 25 will be Ted Goodson. So thank you to them in advance as well for accepting our invitation to present here and to share the work with the community. And also, we would like to acknowledge FaceTech for funding to support this program. And with that, I will pass it over to Lou and for today's introduction.
Cool. Good afternoon, everyone. Welcome to the webinar. I'm very excited to introduce our speaker today, Professor Mark Tuckerman. So Mark obtained his bachelor's degree in physics at UC Berkeley and his PhD degree, again in physics, at Columbia University, working with Professor Bruce Byrne. He then became a postdoc fellow at IBM Zurich, working with Professor Michele Paranello, and then an NSF postdoc fellow working with Professor Mike Klein at UPenn. In 1997, Mark joined the faculty of New York University, and he's now the professor of chemistry and mathematics at NYU. And he's also the chair of chemistry department there at NYU. So I first started reading Mark's work on ab initio MD and path integral methods when I was a postdoc. And then I soon discovered that Mark has an incredibly broad interest in research. So he's not only interested in developing theories in, for example, path integral methods, large time step MD, enhanced sampling methods, and machine learning models, but also there are applications in interesting systems such as liquids, small molecules, biomolecules, fuel cell membranes, and crystal structure predictions, just to name a few. So Mark has won many awards. Just to give you a few of them, these include the Sentinels of Science Award from Problems, the Japan Society for the Promotion of Science Fellowship, the Camille Dreyfus Teacher Scholar Awards, the NSF Career Award, and the NYU Golden Dozen Teaching Excellence Award. It's amazing to me that every time I attend Mark's talk, it's on a different topic, and all of them are very interesting and exciting. So today he's going to tell us about exploring and predicting polymorphs in molecular and atomic crystals. So without further ado, please join me in virtually welcoming Mark for today's webinar. Huge applause, although it's virtual.
It's virtual, right? I got that. Well, thank you very much, Lou, for that incredibly nice and generous introduction. So yeah, as Lou said, I'm going to be talking about phase behavior in atomic and molecular crystals. This was actually a subject of an entire symposium at the last ACS meeting that we had in the spring this year. And some of what I'm going to be talking about is a little bit of an outgrowth of the work that I presented there. I was a co-organizer of that. It was really quite a nice symposium. You can see I have multiple homes. So in New York, I'm here in the Department of Chemistry and the Courant Institute of Mathematical Sciences. So I have to wear both the chemist and the mathematician's hat. And there's lovely Washington Square this time of year in the fall with the pretty colors. And then when it's not pandemic time, I do spend some time also at our Shanghai campus in China, where, well, this is it down here, but there's actually a new campus that's being constructed, a lot larger one. And so probably this year or early next year, everyone will be moving to that one. Well, let me talk about the issue that we're most interested in. And that refers to polymorphism, all right? And that was actually the subject of our symposium, you know, how and in what conditions do molecules crystallize? And so polymorphism refers to the ability of a compound to form multiple crystal structures. And as the title of this slide suggests, you know, when drugs fail, this is one of the things that you can blame on that. So if you think of the drug design process, there's, of course, the compound design and discovery part, and then there's the formulation part, which is, you know, downstream of everything else. And at the end, what you usually do is you need to know, how does your molecule crystallize? From the crystal structures, you usually grind the crystals up into fine particles, you form a tablet, or you create a patch, or something like that, that is the actual delivery mechanism. So and sometimes polymorphism is a real problem in pharmaceuticals. So let's take this example. It's not the usual one that people like to present. They usually like to talk about ritonavir, but I'm going to talk about rotigotine. It's this molecule here, and rotigotine is used to relieve symptoms of Parkinson's disease. All right, so you create a transdermal patch, and like I said, the way you create the patch is you take a crystal structure, you take a crystal form, you grind it up into small particles, and then these particles get spread, in this case, over the patch. The patch is applied to the skin, and then molecules come off of the surfaces of these small particles, and they're absorbed by the skin, and then they do whatever they're supposed to do. So in this case, this is a pharmaceutically active crystalline form, but it turns out that with this molecule, it's polymorphic. And the way you know this is that if you let these patches sit around, you start to see these kind of snowflake-like patterns appear on them. Now that is an indication that there's a polymorphic transition, another crystal structure that's forming, and it looks like this. Okay, it's now called form 2. And it turns out that this is a very stable crystal structure, a very stable form. And as a result, the skin is not able to absorb the molecules from this crystal. And so this becomes a pharmaceutically inactive or biounavailable form. And when this happens all over the patch fails to do what it's supposed to do, and the drug completely fails. And in this case, the drug had to be recalled and then reformulated 4 years later as a kind of amorphous matrix that actually didn't have this problem. Well, you can imagine that, you know, if there's just a little seed of a more stable form and you don't know about it beforehand, this seed gets into the manufacturing process, then you can have conversion of entire batches of your drug going to the more stable form. And if that happens in your manufacturing plant, these little seeds get into the machinery or into the vents or whatever, then, you know, any batch that you create could have this problem. So when that happens, you have to shut the plant down, scrub it clean. And if you still can't get it clean, you usually have to shut the plant down and build another one somewhere else. So there are some estimates that say that there could be 15 to 45% of compounds that have this late appearing polymorph problem, right? You let the thing sit around and you get this conversion to a more stable form. And you'd like to know about this before you go to market with your drug. So you'd like to know, what is the landscape of polymorphs that my compound can crystallize into? Well, doing all the experiments to find all these polymorphs is expensive. It can be time consuming. You don't always know if you've done all the experiments that you need to find these polymorphs. So this is an area where with the right algorithms, computation can actually play and is playing a really important role. But it's not just in pharmaceuticals where you can exploit polymorphism. Turns out that in the area of contact insecticides, exploiting metastable polymorphs actually has an advantage. So this is my kind of analogy of what's happening in the contact insecticide world. So you see a guy walking on these giant crystals in a mine in Mexico, and these are gypsum crystals. And so when you walk on these large crystals, you become what I call a crystal walker. And in the world of contact insecticides, the same idea kind of it works. So you have your crystal, you grind it up into small particles, you spray the particles on the walls of your house or on the plants, whatever, and then the insect will walk on these tiny crystals and they'll absorb the molecules from the surfaces of these small crystalline particles. In this case, they go in through the feet, through the tarsi, and then they go into the insect body and they shut down ion channels and they do whatever they do to kill the insect. Now, DDT, of course, is a long banned insecticide. We don't use it anymore, but in the mid 20th century, it was in use. And the form that was used is called Form 1. And my colleagues here at NYU became interested in, well, what if you had metastable polymorphs of some of these materials? What would they do? And so they made another polymorph of DDT called Form 2. And it turns out that it's a less stable, less thermodynamically stable form, and therefore it turns out to be more lethal. Same compound, you're not delivering anything chemically different, but you're delivering it in a more effective way. And the way you can see that is to do an experiment. All right, so here they took Drosophila and they exposed the fruit flies to Form 1 crystals, Form 2 crystals, and then a control group that wasn't exposed to anything. And as you watch this movie proceed, you'll see that those exposed to Form 1 are actually dying off more slowly than those that are exposed to Form 2. So it becomes clear that the Form 2 is indeed a more effective delivery mechanism for this particular insecticide. And in fact, you can quantify this by plotting what are called lethality curves. So these are fractions of the number of insects that you kill in a certain period of time. And so the 3 trials of Form 1 are in the black, the 3 trials of the Form 2 are in red, and it's very clear that the Form 2 trials are having a faster rise time. That means that you're killing the insects more quickly. This is a more effective crystal form, which is a metastable, less stable crystal form for the delivery of this contact insecticide. Now, as I said, DDT is long banned. We don't use it anymore. So let's go to something that's a little bit more relevant. This is deltamethrin. Right? Deltamethrin is still in use. And the Form 1 is the marketed form. And again, my colleagues made another polymorph they call Form 2. You can see the different crystal structures that you have. And if you look at the lethality curve of Form 1, you can see it's not too bad. These are Aedes mosquitoes. So these are the ones that carry dengue fever, for example. And Form 2 has this really fast rise time. In other words, this is an incredibly effective form for delivering this particular insecticide. Now, the good thing about being able to use polymorphism this way is, of course, you can use less of the material, less of this toxic molecule. And because it's killing the mosquitoes more effectively, they're absorbing the molecules more. Other types of insects that you don't want to be exposed to these molecules, like honeybees, let's say, of course, are in less danger because there's just less of this stuff around. Unfortunately, and as was reported in Science last year, the mosquitoes are starting to evolve to become resistant to compounds like deltamethrin. And as a result, you need to be able to switch compounds quickly. And so another one that we very recently looked at, which is called imidacloprid, and that was published in this JACS paper just this year, and we got a little bit of press on this because This particular compound is incredibly polymorphic, all right? So I think there were 3 known forms, and using this technique of melt recrystallization, where you heat the crystal and then you let it freeze again, and you can cycle through this and create different polymorphs. If you're good at this technique, they were able to get something like 6 new crystalline forms of it. And when you plot lethality curves of some of the new polymorphs they found, for example, 4, 6, and 9. 9 especially, this one down here, is incredibly effective against all the insects that they tested, 3 mosquito types, including the ones that carry malaria, and fruit flies. And form 1 is the commercially available one. And then these 3 polymorphs really have these very fast rise times in their lethality curves. So I think this is kind of a sea change in the way we think about the use or the exploitation of polymorphism as a way to, well, in this case, manage vector-borne diseases, but to achieve a more effective delivery mechanism for something. Now, interesting thing about imidacloprid is that it has these 2 internal dihedral angles that determine the conformational space. And from just a computational point of view, if you imagine, well, what would be the conformation that I would want to study crystallization in? If you plot the gas phase curve from one of these dihedral angles, they're both very similar. The gas phase curve has very distinct minima. And in the crystal form, some of these polymorphs, you see that the dihedral angles are close to these minima, but in a lot of them, they're not. All right. In fact, they're up the walls of this potential energy curve. So what this tells you is that the crystallization, the packing itself, stabilizes what in the gas phase would not necessarily be stable conformations. And this is a challenge when you're deciding, well, what is the monomeric form that I want to use if I'm going to do a crystal structure prediction calculation, such as was used to generate the structures that you see here of these different polymorphs. All right. You need to be able to do this in the crystalline environment. But of course, in order to do that, you need to know what the crystalline environment is. And that usually requires that you've already figured out what the monomer conformation is. So this is a very, very tricky problem. I'm just putting it out there as a challenge. Now, as I said, computation is poised to play a really important role in it. And in order to encourage groups who do computational crystal structure prediction to get involved in this, the Cambridge Crystal Data Center runs every few years a blind structure prediction test. Well, and in this test, they issue a set of targets. The only information that you have are these molecular diagrams. Of course, they know what the crystal structures are, but they keep them hidden from the participants. You take this information, the chemical diagrams you have, and your job is to predict the crystal structures of these things given only this information. So you can see as you look from, the targets are numbered by just sequentially as you go from competition to competition. So the first set of targets were these relatively simple ones. The second one had these, and the sixth one, which was run just a few years ago, you can see the complexity is ramping up, right? And there's now ongoing, we're in the middle of a 7th blind structure prediction test where the targets are incredibly challenging. So what I want to do is just to show you the kind of protocol that we use to do a structure prediction now, and then what we're inventing to kind of make it easier. And so I'll use this circled molecule from the 6th blind test as an example. Okay, so what does the workflow kind of look like, or at least the one that we use? So first of all, you have to choose a monomer conformation. And as I said, that's rather tricky. For this relatively simple molecule, you can optimize this in the gas phase, and that tends to be a really good starting point. And when you do that, in fact, it turns out you see that this molecule puckers a little bit. Then we have to generate a force field. And we've been working with a group of Christoph Shilevich, who I saw is on the call, to do this actually. And they have a nice program where you can give them molecular dimers, and you can do these very high level DFT calculations and you do symmetry adaptive perturbation theory on that. And you get these very nice tailor-made pairwise force fields. All right, they kind of look like this, and they're designed to target one particular molecule. So they're molecule specific. And the reason you want to do that is that with different polymorphs, the energetic differences that you're looking to resolve could be on the order of just a few kilojoules per mole. So you really need very high accuracy to resolve that. Okay, on top of that, you have to start generating random structures, which you do by applying the symmetry rules of crystalline space groups. Do you have to use all 230 of them? I mean, that's a lot, right? 230 space groups is a high number. And it turns out, actually, you don't. There is a kind of rank order to the space groups that appear in the crystal database from the Cambridge Data Center, for example. And so you can look at the frequency at which different space groups appear for molecular crystals. And for example, P21 on C is a rather popular one, P1 bar, P212121, and you can see how the percentages go down. So if you pick, let's say 20 space groups off the top, you're very likely to get the one in which your molecule will crystallize. Okay. Once you've generated some random structures, you energy minimize them using your interaction model. You cluster what you get to find unique structures. Then you can apply an energy cutoff to say everything below a certain energy value we will throw away, we'll keep, everything above we'll throw away. And when you do this, you're doing what's called zero temperature crystal structure prediction. So you're trying to find, a minimum on an energy surface, all right? And usually this procedure can generate tens, even hundreds of possible structures, far more than actually exist in nature. And so it's not as discriminating as you would like it to be. Nevertheless, here's what we get from, this is what we submitted to the 6th blind test competition. So we started with this molecule, we followed the workflow, and what you get out at the end of a CSP0 study is a diagram in which you plot energy versus density. Density, of course, being a measure of how efficiently your molecules are packing. And for the force field that we use for the competition, you can see there's a bunch of structures up here, high energy, low density. And then there's a very few select structures where the energy is low and the density is high. And these are going to be your best predictions, all right? It turns out that our lowest one was not the experimental structure, although it has a kind of motif that does show up, which is due to the puckering. The second one does not. The third one kind of, again, reflects a little bit what you see in the first one, the influence of this puckering. And actually, in this case, the fourth one was the experimentally known form. So you're looking at an overlay of our predicted structure in purple, and the colored one is the experimental structure. You can compare, lattice constants to the calculated structure. This is, of course, done after you submit your predictions. And the best comparison is what they call the RMSD20. So they take 20 molecules out of your structure, 20 molecules out of the experiment, they align them, and then they do a difference of the coordinates. And what we did was we thermalized our crystals at 150 Kelvin, the experimental temperature, using molecular dynamics with the force field. And what you find when you do that is you get an agreement with the experiment, an RMSD20 of 0.14, which turned out to be the best or the lowest RMSD20 of all the submitted structures in this for this particular target. 0.8, by the way, is considered a match. Now, some statistics from the competition. So of all the groups that entered, 21 tried to predict this one. 12 of them got it right, which means that they got it within 0.8 RMSD20. Those who got it within their top 5, there are only 7 such groups, all right? So even a molecule like this is still not all that easy to do. But the fact that we didn't get it as the lowest ranked structure is, something that you worry about when you're going into these. And it would tell you that you probably have to refine the force field a little bit, or you can try to think of the problem differently, all right? And try to bypass the need for a force field. And that's something that we've been working on, all right? A very interesting thing to do. And we were inspired to turn this into a math problem. And since I wear the mathematician's hat sometimes, this is the kind of thing I'm supposed to be doing, right? But we were inspired to do this by the work of a brilliant Swiss mathematician and crystallographer, Johann Jakob Burckhardt, who was at the University of Zurich, and he wrote this really beautiful paper on the history of the discovery of the 230 space groups. Unfortunately, I've only been able to find this in German. I haven't been able to find an English translation. So, but if you read German, this is a really good paper, a really interesting paper to read. Well, basically what he says in this paper, among other things, is that, because group theory describes crystal structures, there should be a kind of a mathematical way, a mathematical theory that would allow you to derive crystal structures based on mathematical concepts. In fact, this is the exact quote from the paper. And if you read German, you'll see what it says. But basically, it's saying that the points in a crystallographic cell is something that you should be able to derive entirely mathematically. So we thought about this. We asked ourselves the question, given the challenge of predicting crystal structures, could we devise a theory, a geometric and topological theory that would allow us to replace a force field and predict crystal structures? Okay, so first we had to ask ourselves, do the known crystal structures obey some mathematical rules? So we took 230,000 structures out of these databases, the open database and the Cambridge database. And we looked at the positions of molecular mass centers and atomic positions and this sort of thing. And we found that these distributions throughout the unit cell, it's not random. It's actually, it's not uniform, and it's rather specific. So what I'm going to show you is just one set of results where we looked at the projection of this distribution onto the BC crystallographic plane. It's like the YZ plane, if you will. You can do this for any planes that you want, and they'll kind of all reveal the same thing. So these are heat maps, distributions of molecular mass centers across this large database. And you can see that for different space groups, indeed, the distribution is not uniform throughout the BC plane, but there are certain regions where you get high probability, or the heat is high on our heat map for all the different space groups that we looked at. Okay, and so this is telling you that there are preferred points where the molecules like to locate themselves. And this is something you can ask yourself, well, are these special points somehow mathematically predictable? And it turns out that the answer is perhaps. What we found is that there is a set of order parameters that can predict these things, all right? They come from the world of computer science, and they're used to do three-dimensional shape characterization, and they're called the Zernike order parameters. And the way you build the Zernike order parameter is as follows. You start with a basis set. In this case, it's just a product of spherical harmonics and radial polynomials, so they have three numbers that quantify them. And then what you do is, okay, so you remind yourself that the way I build a crystal structure is I take a reference molecule and I take atoms in the molecule. And of course, I can generate the symmetry related atoms by applying the symmetry operations of a particular space group. So let's look at an atom I in a reference molecule, and I'll assume that there are Z symmetry related atoms. The way you define the Zernike order parameter is like this. You look, let's say, at a particular atom. In this case, for this coumarin crystal, it's an oxygen atom. Here are the four symmetry related partners of it. You take the center of mass and you take vectors between each atom and this center of mass, and you evaluate the basis functions there. All right, and you sum over all the atoms in your unit cell. That's called the Zernike order parameter for this particular application of them. Now, remembering that the atoms of the unit cell are generated from the one reference atom, the one from your reference molecule, by applying the group generators. The special points that I showed you on the previous slide from these heat maps turn out to be places where the Zernike order parameters are 0. Okay, so if you can solve this set of equations where you can see I've applied the group generators to the reference atom position, and the solution of this will give you the points where that atom should be placed, at least for the reference molecule. And you can do this for each space group. So we call this the ZZP condition, or zero Zernike condition. So let me show you an example to show you that this is indeed predictive. Here's the space group PCA21, all right? And here are the symmetry operators that you can see here. So if I solve those equations for a particular atomic location, oh, you can see that, you know, the solutions come in the form of lines, planes, there are even individual points. And so let's just look at one of these solutions. Let's look at this one. Now, you remember the heat map that I showed you before, it looked like this. And in this case, there's kind of a line of bright color, all right? A line of bright color here. And this is in the BC plane, so that occurs at B or Y is 0.25, 0.75 for kind of any value of C or Z. Well, that's exactly what we're getting. Here's one of the solutions where I have a line of 0.25, Y is 0.25 Z. Here's, I have two lines where Y is 0.75 for any Z, and that's exactly what we're seeing on this heat map. So indeed, these are predictive of where these special points should be. Okay, and here's just an example showing all the solutions, kind of the lines and the planes. The planes, you know, I'm kind of showing them pushed up against the edges of the unit cell, but I'm also showing you here positions of some heavy atoms in my database. These are sulfur atoms, as it turns out, and you can see that these atomic positions are indeed aligning themselves with these special points, with these Zernike or ZZP solutions. Okay, now you can express everything in terms of physical coordinates and physical cell parameters. So for example, if I introduce the cell matrix, which is the matrix of my cell vectors, and the physical coordinates rather than crystallographic coordinates, the solutions of these equations will allow you to find the positions of the reference atom and the cell parameters. So how many parameters do you have to determine here? Well, look, you have to determine the cell lengths and the angles between them, all right? So the basic cell parameters. Then you have to determine the orientation of your reference molecule, the Euler angles. You have to determine where its center of mass is going to lie. And if the molecule is flexible, then you also have internal parameters that you have to worry about, internal dihedral angles and things like that, right? As I showed you, like for imidacloprid. So the total number of parameters is 12 for rigid molecules plus new internal parameters, all right? And so this becomes, you know, a kind of high dimensional optimization or zero searching problem. So let me take you through a little exercise to show you how predictive these things are for a given molecule. So this is coumarin. And what I'm going to do is I'm going to fix 11 out of the 12 parameters. It's a rigid molecule, so there's no internal stuff to worry about. And I'm going to fix 11 out of the 12, and then I'm just going to vary one of the remaining parameters. For example, let's do this for the B, the length of the unit cell in the B direction, all right? Well, I plot the log of the order parameter for a particular choice of NL and M. And as I vary B, you can see that there's a sharp minimum at a value which is 5.6. And the experiment says that it's about 5.6. Let's take a molecular parameter. Let's take the angle theta, the orientational angle, all right? And in this case, for this particular choice of NL and M, there are two minima. So you ask yourself, well, do all the parameters, all choices of NL and M do that? Well, okay, you can do a histogram over a wide range of NL and M values, and you can see that when you do that, one particular minimum really stands out. It's the one at about 90 something. The predicted value is 98. Experiment puts it also at about 98. So indeed, these things are being predictive. So what you have to do here is to then find the structures that are the most fit, the ones that align the atoms and the molecules with these zero Zernike parameters, but that also respect the molecular geometry. And this can be done using a kind of fitness or genetic searching algorithm. There was another thing that we discovered, by the way, which is very useful, which is about the statistics of rings. In organic molecules, many of them have, you know, rings, 6-membered, 5-membered rings. And it turns out that if you want to orient these rings, which is kind of a good way to anchor your molecule when you're doing this kind of searching. If I look at a ratio of the cell vectors, you know, A over B, B over C, C over A, the lengths of these vectors. And I define this such that these ratios are greater than 1. And I compare them to ratios of the components of vectors that are normal to the planes of the rings. Again, defining them so that the ratios are always greater than 1. And I compare the ratio of this fraction to this fraction, subtract 1. It turns out that in all the space groups that we looked at, there's a sharp peak at 0. In other words, this ratio does not want to be different from 1 by very much. Okay, and that's actually another criterion you can use. So let me show you what the workflow of topological crystal structure prediction is. So you start off by generating a lot of structures, 100,000 of them. You choose a density range, usually between 1 and 2 grams per cubic centimeter. Pick a space group, you pick a Z and Z prime value, in this case 4 and 1, which goes, you know, with this particular orthorhombic space group, we pick, you know, the Z and Z prime combination. And because it's an orthorhombic group, you don't have any cell angles to worry about. So it's rigid. So you have 9 parameters to worry about. So this blue cluster here is our initial set of structures. Now, using the ring statistics, or the idea of making this fraction close to 1, you can actually orient these rings. All right, and when you do this and then align everything with the 0 Zernike parameters, you find that there are basically 9 atoms respecting the molecular geometry that align very closely to the ZZPs. And so what we have is a fitness criterion, a delta S, that measures the average distance to these Zernike parameters, all right, of these 9 atoms that want to align there. And so the way you would then pick out a structure to do the next generation of your algorithm is you would find the one with the lowest delta S. And we use that as a way to generate a new set of structures, and we only keep those that reduce the delta S further. And that's the orange set that you see there. Okay, and then once you finally sort of think you're getting close to delta S is 0, you can tweak structures a bit by hand to align them with the ZZPs. And that circled structure at the end is our final prediction. And it has an RMSD20 of 0.1, and it's this structure here, all right? So that is the experimentally known structure of coumarin. Well, it's interesting now to compare the delta S with the volume, all right? Because that, again, is going to give you a measure of efficiency of packing. And so this is a set of structures that we got with different delta S values and volume values. And the one that is the closest match to experiment is this one, with this RMSD20 of 0.1. Now let's go back to the target 22. Remember, our prediction of the force field was this 4th rank structure. But now you do the topological one, and our best prediction is this one, okay? And that's this structure here. And if you compare it to experiment, it's almost as good as what we got with our force field. It has an RMSD20 of 0.19. So we're kind of excited about the fact that we may be able to do crystal structure prediction without actually ever needing to generate a force field. Okay, but this is not the only problem that you have. As I indicated in the beginning, the other thing that you want to be able to do is to predict the kinetics of polymorphic transitions. All right, and we haven't extended the topological theory into that realm just yet, but we are doing this with, you know, physics-based approaches where we have force fields and all that. Hopefully we'll eventually get the topological thing to work there too. But so my kind of illustrative example of the drama that you can have with polymorphic transitions, it's not pharmaceutical and it's not contact insecticides, comes from the lab of my colleague Panche Namov in NYU Abu Dhabi. And he makes these crystals where if you heat them, you can actually get a polymorphic transition as a function of temperature that releases a lot of energy. These are called thermosalient crystals. And he has some samples here that he put on a hot plate. And as you heat them, you get this polymorphic transition and they jump off the plate. All right. So these are called jumping crystals. And it's thought that the way that this happens is that you set up an interface between two polymorphs in your crystal. The interface migrates through the crystal and causes the transition to go layer by layer, each one releasing more and more energy. It builds up, and then these things jump off of the hot plate. Okay, so you can imagine a kind of interface and where I have two structures that come together, and that interface will migrate through the system. Well, we wanted to do this, but we thought starting with molecules would be a little bit too hard. So we thought, let's start with something simpler where we can hone the techniques, and then we'll see how we can adapt that for molecular crystals. So we started with a simple atomic crystal, in this case, molybdenum. Now, molybdenum has a number of phases, but there are two that were particularly interesting to us, which is the so-called A15 phase. That's the one shown in red over here, and that's the one that we use for spintronic memory devices. And then, of course, the more traditional BCC phase, which is used for electronic applications. And the A15 is a kind of exotic one that you don't see in all atomic crystals, but molybdenum does have it. A15 is known to be metastable. So if I just run a molecular dynamics calculation, I should eventually see the A15 convert to the BCC, okay? But it takes a really long time on the time scale of my simulation. You can see that I'm running this thing, many nanoseconds, and it's just not happening on any kind of, I get a few transitions, but I'm not really getting good statistics on the event. And what's the reason for that? The reason for that, if you do molecular simulation, is known to be the fact that the energy landscape is a rough object where you have many minima that are separated by barriers. It can be high compared to KT. And the way you would usually then try to sample states on this surface is to use enhanced sampling methods, all right? Most of them are not efficient enough to handle this kind of polymorphic transition. Even the ones that use collective variables, you just don't know what the right collective variables are to describe this transition, all right? So direct simulation of the transition is difficult. So what we decided to do is to turn to machine learning to help us figure out how these transitions go. And so as I go through this, I hope you get a sense of how the machine learning works and why we're using it. So the first thing that you might do is to identify all possible phases, all right? And so those I might be able to get by doing an enhanced sampling calculation. I can generate a free energy landscape. It'll tell me where the basins are, but it won't tell me anything about kinetics, all right? So if I get a landscape like this, it show me the different possible crystal structures. In this case, because it's an atomic crystal, you have BCC, FCC, HCP, A15, that kind of thing. Then I want to be able to generate transitions between them. So if I want to use machine learning, I have to have a set of descriptors that can describe these phases, all right? So how can I distinguish one phase from the other? Well, in order to do a machine learning training protocol, a good set of local descriptors are the same ones that one uses when you generate neural network potentials, all right? These are what are called symmetry functions. They're used because they're translationally and rotationally invariant. And what they do is they describe the local environment of a particular atom. So let's say I have an atom at position Ri. Then these symmetry functions can be, say, Gaussians of different width at different locations that are then going to pick up, which neighbors, as I, search over neighbors of I, which ones are in the neighborhood of the one that I'm looking at, all right? And I can pick up different shells using these. I can also use these cosine ones, which, can assign, let's say, a positive or negative sign to atoms in different environments. So if I want to distinguish, what's maybe first and second solvation shell, that kind of thing. And then these cutoff functions determine the locality or the nearsightedness of these descriptors. These are distance-based ones. So I may supplement this with, for example, Steinhart order parameters. They're a little bit similar to the Zernike parameters. They're sensitive to angular information. So let's say I have a central atom and I look at the neighbors, and what I would do is I would take vectors from the central atom to its neighbors and then look at the atoms, the angles of those between those various vectors. And that's how you construct a Steinhart parameter. Okay, so these are all good local descriptors. Our goal then is to train a neural network to classify phases based on this local structural information. So what we're going to do is we're going to train a neural network to classify different phases. All right, so you come in with a full set of atomic coordinates, you turn them into symmetry functions and Steinhardt parameters, and then you run them through the layers of a neural network with your edge weights that will be the parameters you train. And you'll come out with a set of probabilities that say this atom has certain probability to be in an A15 environment or a BCC environment or whatever, right? And since they're probabilities, if you sum over all the phases, they have to sum to 1. And so you can see I have 4 phases and then general disordered or liquid-like configurations that don't have any order to them at all. So once you have your local descriptors, then you turn them into a global descriptor by averaging these. And that gives you a descriptor for what's in my simulation cell. So here's a snapshot showing some BCC, some A15 and interface. It's got some other stuff in there. And what you can see is that over here, I've colored the BCC as gold. And my local descriptor, my global descriptor says, okay, I've got about 0.2 fraction of BCC. If I color the A15 in gold, then you can see that my global descriptor says, okay, you've got about half of that as A15. And then these don't sum to 1 because you've got other stuff in there at the interface. All right, and you have to classify that too. Okay, so how will you train this neural network? So the way we do that is we generate a lot of atomic environments, either just pure bulk phases or also mixed phases, all right? And we're going to do that by running MD in pure and mixed phases at different temperatures and just extract lots of configurations. And since I know what the environment is, I can train my network this way. And for the mixed phases, I have to use enhanced sampling methods that can generate these mixed phases because they don't, appear all that easily by themselves. And of course, when I use my descriptors and I drive the simulation this way, I can actually generate the free energy surface, right? And this is the free energy surface, not the 14-dimensional one that has all these descriptors, but I projected it down to 2 dimensions, all right? So I generate the 14-dimensional surface, but I project it down to 2 so you can really see where all these phases are. Okay, so once you have mixed and bulk phases, then you train your neural network. The architecture that we use is 2 hidden layers with 25 nodes in each layer, your 14 input nodes, and then of course your 5 output nodes. Okay, and because it's a classification problem, our cost function is just this cross entropy function. So my neural network form goes here. These are the target values of my classifiers. And then to test it, you generate another set of 125,000 different atomic environments. And then you can use that to validate your network and generate these things called learning curves. So the learning curve is plotting the error, your cost function, versus the number of training points that you had to use to get that error. And so you can see that for everything but the cyan, all right, so these are all my bulk phases, training is very fast. With a few hundred training points, I basically got the error down to 0, all right? For the mixed phases, of course, it's mixed phase, so it's a little harder to classify. But again, with a few thousand, maybe 10,000 points, you see this curve is leveling off. And it doesn't go to 0 because it's never easy to get perfect classification of interfaces. But you can see that we got it to about 93 to 94% accuracy. And that is actually very good for a classification neural network. Okay, now that we have the network, what do we do with it? All right, and this is where we're going to devise a collective variable that can be used in an enhanced sampling calculation to drive the transition. All right, what will we do? How will we do that? So what I'm going to do is I'm going to take the classifier, the global classifiers of the 2 phases I'm interested in, and I want to build a path from one to the other. So I'm going to be working in this classifier space. All right, and what we're going to do is we're going to build a kind of path collective variable in the classifier space. All right, so the QIs here are a set of nodal points that, let's say, march from the A15 to the BCC phase, and this Q of R is what my neural network will spit out as a function of your atomic coordinates, what a given environment classifies to. Okay, now this collective variable is designed to have, a different value at each node along your path, and so it can distinguish the endpoint phase as well as things in between. And when you run this when you drive this in a simulation as a collective variable, you're basically going to be generating different paths with these nodal points from one phase to the other, such that you're sampling low free energy pathways. All right. So it's like, moving along these foliations to generate different pathways. Okay. Now, in order to ensure that the sampling doesn't get too crazy, you can put a penalty on how distorted the paths get. That's done with this kind of variable here. It kind of builds a sort of bowl or a wall that says you shouldn't morph the path too much, or if you do, you get a penalty for it. And you can use this as either a second CV, collective variable, or an external bias. And the point is that once you have these collective variables, you can use them in an enhanced sampling approach like umbrella sampling or metadynamics or adiabatic free energy dynamics as a way to generate your paths. So I'm not going to go into the details of how we do the enhanced sampling, but let's just see what our simulation looks like when I drive this path collective variable. And you can see that it's now quite easy to generate the interface migration. Okay, so we readily convert the A15 to the BCC phase, but you can also grow the A15 back in. All right, so I can get statistics on the interface migration problem. And that's enough for me to generate an actual free energy profile. So let's say I do this at fixed volume, all right? And when I generate, or I get the free energy profile out along my path collective variable, you can see it's telling me a lot of things about what's going on. For example, you see these bumps, all right? And each bump has about the same height of about 0.5 EV. That is the free energy barrier to convert one layer from A15 to BCC. It's about 50 kilojoules per mole. So I don't have an experimental number to go with this because the simulation cell is too small, but there was a kinetic Monte Carlo study done some years ago by my collaborator, Dr. Jutta Rogel, who showed that, within their scheme, it costs about 0.5 electron volts to go to change one layer from A15 to BCC, to have this transition, this solid, local solid phase transition, all right, one layer to the other. But it's also downhill, all right? And the reason that you don't see that here is that we're at fixed volume. But if you do it at fixed pressure and you let the volume vary, then you can see that the profile you start with, which, this is what we had after just a few nanoseconds, as it evolves, it starts to have this downhill kind of Galton staircase-like structure to it that's showing you that it is indeed downhill from A15 to BCC. That's illustrating the metastability of the A15 phase. But there are still barriers as you go down, and they're, about 0.5. They still retain this kind of 0.5 EV value, all right? And so, that tells you that A15 is certainly metastable enough, as long as you don't go to too high temperature, that you can use it for things like, the spintronic memory devices that they make. Okay, so my last slide, and I'm just going to finish up by showing you how we will turn this into a scheme you can use for molecular crystals. And molecular crystals, it's not just about the distances between molecules, but you need some additional symmetry functions that tell you about orientation. So we're going to use this thing called the point vector representation. Let's suppose I have a rigid molecule like urea, and I put some point vectors that span from, let's say, a carbon to an oxygen or a nitrogen to a nitrogen, and they can describe different ways the molecule can orient. Then between neighbors, you get the cosine of the angle between them, and you build the same kinds of symmetry functions as we had before, but they're functions now of the angles rather than of the distances. And if I drive these collective variables just by themselves, no machine learning or anything, in an enhanced sampling simulation, let's say of urea, this is urea, you can see that, in fact, I can move these things easily from one polymorph to another. In other words, I can generate different transitions. This is not getting kinetics. This is just exploring the free energy landscape. All right. But these descriptors can distinguish the different polymorphs. Let's look, for example, also at another fun molecule. This is resorcinol, where you actually have a conformational dependence of the polymorphism. So there are a number of different conformations for these OH groups that dangle off the benzene ring. And it turns out in the gas phase, this middle one is the lowest energy conformation. But in crystal structures, that's no longer true. All right, so here's a landscape of polymorphs of resorcinol. And the three polymorphs that you see up here, two of them are known experimentally, the epsilon and beta. They use this middle form. All right, so you can see that in the beta form, let's say this one, the epsilon form, this one, the OH groups are in this trans-like configuration, but in the alpha phase, which is this phase here, the two OH groups are in this cis-like configuration. All right, and so like a metacloprid, like we had before, the gas phase minimum is not necessarily what's stabilized in the crystal structure. And so if I drive these symmetry functions, I can actually get the beta phase or any one of these higher energy polymorphs to convert to the alpha phase. And in doing so, you have to get this internal conformational change. All right. Again, I'm not getting the kinetics right here because I haven't used the path collective variable, but at least I can explore the free energy landscape and see, what structures I would need to include. Of course, I already know them from my diagram, but if I didn't know that, this would give me a set of phases that I should use. And then, of course, the next step, which we haven't done yet, is to take all this information, put it into our machine learning protocol, work up with these path collective variables and see if we can get the kinetics of the transitions, right? Using different types of interface setups, local seeds where the interface may be spherical, putting them on, at surfaces and seeing if the transition happens more readily at a surface than in the bulk. This kind of thing is what you'd want to do. Okay, so I can see I'm out of time. Let me just wrap up with a few conclusions. So the last thing I showed you was that, you can use machine learning, you combine it with molecular simulation and enhanced molecular simulation to produce kinetics of polymorphic transitions. In the atomic crystal case, we would like to extend this to molecular crystals. I consider that one of our holy grails. We haven't quite done it yet, but it's on our to-do list. And earlier in the talk, I showed you that you may be able to bypass force fields in predicting crystal structures using just the mathematical properties of crystals that we know from these databases and the topological theory. I showed you some simple predictions for small molecules. Of course, we acknowledge and understand that the road ahead is long to be able to do flexible molecules, crystals with high Z prime number. I mean, the really challenging cases are still out there. So the road ahead is still quite long. Okay, I'm going to finish up. Let me thank my collaborators on this. So all the topological work was done by a very brilliant mathematician postdoc of mine, Nikos Galanakis. The work on the atomic molybdenum crystals was done by Dr. Yuta Rogel, who's in my group now as a Heisenberg Fellow. The work on the blind test was done by Leslie Vogt and a former postdoc of mine, Elia Schneider, and a student, Hongxing Song. We work with Christoph Shalevich on the force fields, as I described to you. And of course, my experimental collaborators, Panche Naumov, Mike Ward, and Bart Carr, who have been challenging us with different molecular crystals that we can look at. And please follow us on Twitter if you're there. Otherwise, thank you very much for listening to my webinar, and I look forward to taking any questions that you have.
Thank you, Mark, for a very interesting talk. I see there are a few questions from our audience, and we do encourage our audience to keep typing your question in the chat, and we'll ask them. All right, the first question we got is from Gregory Barron. Regarding the topological structural prediction, what happens if you look at a set of experimentally known polymorphs? Do they cluster in the optimal region of the landscape? Alternatively, could mapping a traditional energy-based CSP landscape onto the topological landscape help eliminate spurious structures that are unlikely to be realized experimentally?
So both good questions. We have looked a little bit at polymorphs. We looked at the known polymorphs of coumarin, for example, and it turns out that if when you look in the right space group and all that, these structures do actually appear as the structures that within that space group follow the topological rules. In other words, they align themselves with the zeros of the Zernike parameters, and they stand out that way. When you look at the wrong space group, you actually find that there are no physical structures that you can get. Now we haven't looked at lots of examples in order to kind of confirm that this is a reproducible fact, but for the ones that we have looked at, this does seem to be true. So yes, the answer is that the polymorphs do kind of stand out. And when there are multiple polymorphs in a given space group, they usually sort of show themselves as like structures where you say, okay, yeah, that one obeys the rules. This one obeys the rules almost as well. And they sort of stand out from the rest of the herd. And so you'd say, oh, there are 2 structures or 3 structures that kind of obey the rules. We would predict that these would be polymorphs. The other one that you were asking, the other question was about combining this with energy evaluations. And of course, we're trying to get away from energy evaluations. But I think, yes, it perhaps could, because it could help you eliminate spurious structures, because it would give you a way to say, okay, look, if my topological theory predicts some structures, but then these structures end up having too high an energy or the overlap is too much, yeah, they would have a physical signature like that, a high energy or some other thing that you would say you would probably want to throw them out. So there could be room for doing that. It's just that that's not our goal. Our goal is to try to do structure prediction without having to go through the step of generating a force field, which is, it's time consuming and it's resource consuming. And, let's say in the blind tests that we've done, it really does take a full year to get a force field generated and then do the structure prediction. It can take weeks to months, right? Whereas when we do this with the topological theory, it's a matter of just hours to do one structure prediction, maybe days if you look at lots of space groups. All right, so we're saving a lot of time by bypassing the force field step.
Before Marco asks the second question, I want to remind the students that after the talk today, we have 20 minutes of questioning time with Mark. So you're encouraged to participate in that session.
Okay, the second question is from Qianzhang I hope I didn't mispronounce it too badly. So the question is, for the slide with heat maps of a central mass of molecules in unit cells, the structures are translationally invariant. If I translate every atom for every unit cell in CSD by, say, 0.1, 0.2, 0.3, would that change the heat map? Would this affect the following ZOP conditions? And also, if you could share the reference for this work.
Okay, we don't have a reference yet. So this is as yet unpublished work, which is what I like to present usually, because the published work you can go read about. But yeah, so your question, of course, is of course you have translational invariance, and you can shift everything would shift where the zeros are. We could also do the same thing with our Zernike parameters, but keep in mind that the Zernike parameters are generated using relative vectors, right? Remember what I said is that you take all the symmetry related atoms, you find their center of mass, and then you construct the relative vectors between them. And that's what we're aligning, right? So that's what we're solving for. So it does, the translational invariance doesn't actually matter. Maybe I go back to the formula. Not that one, this one, right? And this shows you that there is no absolute, we're not working with absolute positions. Everything is coming from these relative positions. So in terms of what we found for the heat maps, yeah, that may be true, but what we're actually predicting doesn't really care about where your unit cell is. But there are conventions, of course, that we follow for how to put the molecules in the unit cell. And so we do follow that. But the Zernike parameters don't care.
Okay, our next question is from, I hope I get your name right, Chamikara Karunasena. Okay, maybe I'm not too bad, but so this question is why do you look into the lowest free energy path to output the best collective variable? The best collective variable should be the one describing the highest energy associated with transition or the slowest mode of transition.
We think we're getting that. I mean, what you're asking about is, are we getting the barrier right? Right. So when we say the minimum free energy path should be one that, would take you over a saddle point. You'd hope it would be the most accessible saddle point or the easiest one to cross. But at the end of the day, we know that the path from one structure to another will go over a barrier, will go over a saddle point. And we seem to be getting that. The reason that, maybe, I don't know if this answers the question a little bit better, but the reason that we are working in this classifier space is that if I have to go over, if let's say I have to go through a series of structures that will cross a saddle point, surely, from one structure to another, and I want to construct a set of nodal points on a path. If I do it in configuration space, then I have to decide what those nodal points are. In other words, what those configurations are. And that means I put in my conception, my bias about what I think those configurations will look like. All right. And I don't want to do that because I have no idea. So if I work in the classifier space, all I have to do is to say, look, I'm going to march from the classifier of phase 1 to classifier of phase 2. And that's an easy path to set up. And then the machine learning tells me what configurations I have along the way. And that should give me the right set of configurations along the path, including whatever barriers are there, as I go from one phase to the other. And we do seem to get the barriers right. I'm not sure if that's answering the question, but I think hopefully it is.
Okay, the next question is from Jaron Holt. Question is, why do molecular structure change to the one or lowest energy when near ones already are in that sort of state? I think is referring to how do you get this phase transitions to get to the lowest energy polymorph? If you can clarify that.
Okay, I'm not entirely sure I'm understanding the question, but I mean, my understanding is that, structures, they may be close in energy, but they're not necessarily close in terms of the actual structure themselves. So as I see it, you are going to rather different structures And that's why it's difficult. There could be large barriers between them. Otherwise, if the structures are similar, let's say you have that situation, and there may be, like one molecule orients a little bit differently, or let's say you have a stacking fault or something. Okay, that can happen. Usually we think of those as maybe intermediate structures or polytypes, or things that are not necessarily what you're looking for. They could be if you have a reason to, but what you're really looking for is a very different structure, which will have different properties. I'm not sure if that's answering the question, but it's the best I can understand it.
Our next, I'll wait for this question then. On the list, my next question actually comes from Marco, so I'll wait for Marco to ask the question.
Okay, I'm gonna ask my question directly in the meantime that we get more questions in the chat. So my question was, if you can take into account multiple conformers of the same unit molecule within the same crystal structure, especially during a phase transition.
So I think the answer to that is yes. I mean, if we're doing structure prediction and there's that kind of disorder, then we would need different techniques. But if you're looking for this kind of thing coming up during a phase transition, like where you have one structure with this one conformer and a structure where you have, you're going to a structure with this different conformer, I think you can actually do that because you've trained your neural network, right? You know what the endpoints are, and intermediate to that, you don't know, but you're going to be, driving the thing from one to the other. I think you should be able to do it. For example, when I looked at resorcinol, it's admittedly very simple example, right? But you saw that as I went from the beta polymorph, where the OH groups are like this, you can't see my arms, but anyway, and then when they're like this, right, you were able to get that structural transition. Now that happened naturally. Whether that's going to happen when the barriers to that transition are high will probably depend on how we do the enhanced sampling. For example, it may not be enough that you have the path collective variable. You might have to throw in some internal dihedral angles into your enhanced sampling in order to get it. You'd like not to, like just to be able to have it go naturally. But that's something we have to explore. But I think, with the right protocols, if it's supposed to happen, it should happen.
Well, our next question is from Tucker in the RISCO group at University of Kentucky, asking, have any structure more complicated than molybdenum being put through the machine learning model yet? Likewise, how was overfitting avoided for this model?
Okay, so the answer to the first question is not yet. We have started to look at urea crystals as like our first simple molecular crystal, and we've been able to train the neural network on, I think we had four different polymorphs we were looking at for the force field that we were employing, and so we can train. But what we haven't done yet is to generate the path. So that's yet to come. Overfitting. Let's see. How do we avoid overfitting? I mean, it's a classification problem. Let's say it this way. I mean, we trained a bunch of different neural network architectures to see if they would kind of give the same classification accuracy and classify the training sets in about the same way, and they were able to. So I don't know if that rigorously gets to the, how do we avoid the overfitting, but we're able to at least verify that different architectures were able to do it. We just picked the one that had the most efficient use of the training data. So not a perfect way to avoid overfitting, but a way that we could show at least the robustness of our training protocol.
Well, very, very great discussions, but in the interest of time, we probably should switch to the student and postdoc session. So we'll ask the professor to leave. Thank you for coming. And then we'll have a discussion session with Mark.